Animal Kingdom, the fourth chapter in the PUC first year biology. Now we have already studied about classification. We are following R.H. Whitaker's classification, and according to this system of classification, the living organisms are classified into 5 kingdoms: kingdom Monera, Protista, kingdom fungi or Mycota, Plantae, and Animalia or animals. So in the second chapter we have studied the first 3 kingdoms, that is Monera, Protista and fungi, the characteristics and a brief classification and examples of that. And in the third, that is plant kingdom, there we have studied the groups under plant kingdom like algae, bryophytes, pteridophytes, gymnosperms, angiosperms. Now the animal kingdom, which is the 5th kingdom according to Whitaker's classification. So R.H. Whitaker, who classified living organisms into 5 kingdoms, so the kingdom Animalia or any kingdom is further divided into divisions according to the taxonomic hierarchy or the Linnean hierarchy, which we have studied. So the kingdom is further divided into divisions or phyla, single phylum. Then division is further divided into class, and class into orders, order into family, and each family is having the genus and species. So animal kingdom, it again consists of divisions or phyla or phylum. So we are studying different phyla in this animal kingdom. Mainly animals are broadly classified into invertebrates and vertebrates. So there are several phyla in the invertebrates, and also there is a single phylum in the vertebrate, that is phylum Chordata. Then before going to the actually or the number of the phyla or the characters of each phylum with examples, there are some basic terms which are related to the phyla or the classification. Now what are those terms we shall study first. So here, complete digestive system, incomplete digestive system. So open type and closed type of circulatory system. This is one classification. And again asymmetrical, then radial symmetry, then bilateral symmetry, then diploblastic, triploblastic, then triploblastic, then coelom, coelomates, pseudocoelomates and acoelomates. So these are some basic terms. So acoelomates. Then other than this, we are having another 2 terms, metamerism and notochord. So these are some basic terms which are related to animals, whether it is a invertebrate or vertebrate. So what are complete animals which are having complete digestive system? So complete and incomplete, these are related. So open and closed, asymmetrical, radial symmetrical and bilateral symmetrical, diploblastic, triploblastic, then coelom, coelomates, pseudocoelomates and acoelomates, and these two are separate individual terms. Now what is complete digestive system first? So a digestive system which is having a mouth and anus, so an opening through which the food and water enters into the body, which we generally call as the mouth, and the undigested food material after digestion inside the body of the animal, so it is sent out through another opening which is called as the anus. So an animal which is having two such openings like mouth and anus is called as complete digestive system. So or that digestive system is said to be complete. Now when there is only a single opening, it is incomplete. So the entry of the food for digestion, it is the mouth, and the undigested food which is sent out through the anus, that is the another, it is complete. So there are two distinct openings for the entry and exit of the undigested food material. Now in incomplete there is only single opening, so through the same opening the food has to enter and the undigested has to come out through the same opening. So usually in some animals like Platyhelminthes and even in the Coelenterata group, we have come across such examples, that is complete and incomplete digestive system. Then open and closed type of circulatory system. So this is related to circulatory system. So the blood which is present in the animal, so when it does not flow in well defined vessels, it is present in the body cavities where the blood, the organs, the body organs are embedded or immersed, in other words, in that fluid, that is the blood. It is called as open type of the circulatory system. So the blood, in case of insects, there the blood does not flow in through well defined vessels like arteries, veins or like that. It is called as open type. So it is present freely in body cavity where the organs are surrounded by the blood, that is open type. Now closed type means where the blood flows in well defined vessels. For example, we are the example, human being. So the blood in our body flows in well defined blood vessels which we call as the arteries, veins, capillaries and all that. So that is closed vascular system. Then coming to the symmetry, asymmetry, radial symmetry and bilateral symmetry. Now in case of an animal, if you cannot divide the animal into two equal halves through its central axis, so when an animal is taken, we cannot divide that animal into two equal halves in any plane, either in this way or this way. However you divide, in whichever direction or in whichever plane you divide. So when you cannot or we are unable to divide that or cut into two halves, equal halves, it is called as asymmetrical animal. So the sponges which belong to the phylum Porifera, so they are asymmetrical, where in whichever direction, in whichever plane, we cannot divide the animal into two equal halves. Then radial symmetry means we can divide the animal into two equal halves through any plane. So in any plane we can divide the animal, and that is called as radial symmetry. For example, the starfish of phylum Echinodermata or any Echinodermata animal, we can take starfish, brittle star or like that. So that animal can be divided. So if the animal is placed there, if we can divide the animal in any plane, either in this direction or in this direction or in this direction or in any plane through its central axis, it is called as radial symmetry. So radial symmetry is the animal can be divided into two equal halves. It is in through any plane, it is called as radial symmetry. So equal halves in the sense means one half is the mirror image of the other. So that means when the two halves are divided, so whatever organs are present in one half, the same organ should be present in the other half. That is called as a mirror image, we call it as. So when one half is the mirror image of the another half, then it is said to be equal halves. So symmetry is taken into that sense. So radial symmetry we can divide into two equal halves through any plane. So when the animal is divided into two equal halves through only one plane, it is called as bilateral symmetrical. So suppose an animal is there. So if I divide this animal in this direction, I get two equal halves. If we divide in this plane or in this plane and this plane, we cannot get two equal halves. So the animal can be divided into two equal halves only in one plane. So that is called as bilateral symmetrical. So this earthworm is an example of bilateral symmetrical. So this is about the symmetry of how the body can be divided into two equal halves or not. Then diploblastic and triploblastic. These are two terms which are related to the arrangement of the layers in the body wall. Organisms, especially these layers are clear or they are clearly distinguished in the embryonic stage. So in the embryonic stage of any animal, if we have two layers, so di means 2, tri means 3. So on that basis we can find out whether it is having 2 layers or 3 layers. So diploblastic means there are 2 layers which are referred as the ectoderm and endoderm. So the outer layer is the ectoderm, the inner layer is the endoderm. But in between the ectoderm and endoderm, there is a non-cellular layer which is called as the mesoglia. So mesoglia is not made up of cells. It is a kind of some gelatinous or jelly-like material which is present in between the ectoderm and inner endoderm. So this diploblastic means actually there are only 2 cell layers, but there is also another middle layer, but it is not a cellular layer, it is a non-cellular layer which is gelatinous or some jelly-like substance which is in between the ectoderm and endoderm. So diploblastic. So the lower invertebrates like Coelenterata and Ctenophora phylum, they are all diploblastic in nature. That is what is diploblastic. Then if we come to triploblastic, 3 layers. So there was only ectoderm and endoderm. Here there is one more layer in between ectoderm and endoderm. We refer that as the mesoderm. So these 3 cell layers, ectoderm and mesoderm, middle and inner endoderm, they are very clear, which can be clearly differentiated in the embryonic stage. But all higher animals, even if you take vertebrates and higher invertebrates, they are all triploblastic. That means even we are also triploblastic. Means when we are in the embryo stage, there we find 3 layers in the embryo stage. The outer ectoderm, the middle mesoderm, the inner endoderm. All these are 3 cell layers. And as the animal grows or the embryo stage develops into the young one and further into adult, we cannot see these 3 layers. However, they are not disappeared. The layers have given rise to, they have developed into certain organ systems. For example, if we take the human being itself, the ectoderm, when we were in the embryo stage, it gives rise to the entire nervous system and this skin. So the entire nervous system is developed from the ectoderm. And similarly, the mesoderm gives rise to the muscular and other systems. The endoderm also gives rise to the other systems. So like that, they are developed into well differentiated organ systems, and they are called as triploblastic when there are three layers: ectoderm, mesoderm and endoderm. So this is what is the diploblastic and triploblastic. Then coelom, coelomates, pseudocoelomates, acoelomates. Coelom is a cavity in the body of an animal in between the body wall and the gut region, that is the inner organ systems. So in between the space which is present there, that is the body wall and the gut system or the gut region, and this cavity is lined by a layer called as mesoderm, what we have come across in the triploblastic. So that mesoderm is present, or it is lining the cavity of that coelom. So the coelom is the cavity in the in between the body wall and this gut region, and it is lined by the mesoderm. That is very important. Simply the presence of cavity is not coelom. It should have the mesoderm layer. They are called as this is called as coelom. And animals which are having this coelom, they are called as the coelomates. So this coelomates are animals which are having this cavity, and the presence of mesoderm, which we call it as coelom, it is usually referred as true coelom. That means mesoderm. Now sometimes the cavity is present but there is no mesoderm. Such type of coelom is called as pseudocoelom. They are called as the pseudocoelomates. So in coelom also there is cavity, in pseudocoelom also there is cavity, but there is only one difference between the coelom and pseudocoelom. In coelom the cavity is lined by the mesoderm layer, but in pseudocoelom the cavity is without mesoderm. So that means the mesoderm is completely absent in case of pseudocoelom. Then acoelom means the coelom is completely absent. So there is no question of mesoderm and that. So the cavity itself is completely absent, and they are called as acoelomates. So acoelomates, pseudocoelomates and coelomates. So most of the higher animals are coelomates, and certain lower organisms like roundworms and all, they are coelomates. And acoelomates means they are also certain lower animals, invertebrates, they are acoelomates. So coelom is the cavity with the mesoderm. Animals having that is called as the coelomates, and usually it is also referred as true coelom, or sometimes it is referred as eucoelom, EU, or only coelom is also referred. And when the coelom is present without mesoderm, pseudocoelom, when the coelom is completely absent, it is called as acoelom. So acoelomates. So like that, the body cavity related to these are the four terms. Then coming to what is metamerism and what is notochord. So metamerism means it is a segmentation of the body. So when these segments or when the body is externally and internally divided by segments, we call it as metamerism. Suppose when the body is there, on the upper side you will find some segments like this, but inside you don't find segments. It is not metamerism. Now for example, in case of cockroach, commonly we see just by removing the wings of the cockroach, or just below the wings, when you see the part of the abdomen, you can see the segments in that body. So those segments are only externally. but the body is not divided by segments internally. When we open the abdominal part just by dissecting, we come to know that. On the upper surface you can see some lines, so that means they are segments externally only, but not internally. So that is called as this is not metamerism. But in case of earthworm we find this. In case of earthworm we find segments not only externally but also internally. The body is externally and internally divided into segments. Such segments are called as true segments, and this phenomenon we refer it as metamerism. So metamerism means the presence of segments where the body is divided internally and as well as externally. We call it as metamerism. Then notochord. Now we have already referred the triploblastic and diploblastic. In the diploblastic, of course, there is no mesoderm. There is ectoderm, endoderm, and in between there is non-cellular layer called as mesoglia. But in triploblastic we find 3 cell layers: ectoderm, mesoderm and endoderm. So this mesoderm, in case of triploblastic, in the embryonic stage, suppose not always, in only higher animals we come across this. So this mesoderm develops into a rod-like structure in that animal. When it develops into a young one at adult stage, then that structure is called as notochord. So the rod-like structure which develops from the mesoderm in the embryonic stage. In the embryonic stage itself, it refers or it develops at the embryonic stage, and that is called as the notochord. So however, the notochord further develops into in case of vertebrates into the spinal cord and that, but the notochord is seen only in the embryonic stage developed by the mesoderm. So that is about the terms which we have come across before going to actual classification. Now we have to study what are the divisions which come under kingdom Animalia. So kingdom Animalia is divided into various phyla. So first we shall study some invertebrate phylum or phyla. So phylum Porifera is one such phylum, a primitive phylum. Then after that, phylum Coelenterata we study. Then phylum Ctenophora. So these are the primitive phyla, phylum plural phyla. So Porifera, Coelenterata, Ctenophora, Platyhelminthes, then Aschelminthes, Annelida, and so on. Now phylum Porifera. Now actually in the old classification, before phylum Porifera, Protozoa group was included. The protozoa what we study under kingdom Protista. So when we study under kingdom Protista, we remember about the chrysophytes, dinoflagellates, euglenoids, slime molds, and the protozoa. So these are the groups what we have come across in that Protista. But Protista was considered as a phylum of animal kingdom. So that means the examples which we come under phylum Protozoa: amoeba, entamoeba, paramecium, euglena, plasmodium, and various others, Vorticella, Daphnia, and many others. So all those which are come under Protozoa, they were considered as animals. But when further studies were done, they were thought to be organisms which were related to animals but not true animals. So that is why they were separated from animal kingdom and included under kingdom Protista by Whitaker. So R.H. Whitaker, according to his classification, protozoa comes under Protista. But other taxonomists who have classified, they consider protozoa as one of the phylum under kingdom, animal kingdom. But here we are studying protozoa under Protista. Then phylum Porifera is the primitive phylum of animal kingdom. So phylum Porifera are primitive animals. They are multicellular. So in animals, when we study, all are multicellular. There is no any unicellular organism coming under each phylum. So these are multicellular, but we don't come across any such kind of organ systems, as there are different systems, but such kind of systems are absent in case of phylum Porifera, because it is a very primitive animal. And these animals, they are generally marine and they are also found in fresh water. So both the types of animals you come across in this Porifera, and usually these animals are sedentary. So that means they are usually attached to some hard surfaces like rocks or any other hard substratum. So Porifera, the word itself suggests, it is actually a Greek word. Por means a small hole, fera means to bear. So these are pore bearing animals, and these animals which are present, so suppose this is a part of the body of a Porifera, they are commonly called as the sponges. So they are attached to substratum just like rocks. The body is asymmetrical again. So when we studied about the terms, so we had given the sponges as the examples for asymmetry. So sponges are asymmetrical. We cannot divide the body into two equal halves in any plane. And the important character of this phylum is water transport system. So this water transport system is an important character of this phylum, which we don't come across in any other phylum. It is also called as a canal system. So water transport system or canal system is the characteristic feature of phylum Porifera. So the body wall is made up of different layers, but here we don't come across diploblastic or triploblastic. So that is there are no mesoderm, ectoderm or like that. And so here the body wall is having minute pores on its surface. So these minute pores, they are actually called as ostia. Ostium single, ostia plural. So on the surface of the body there are ostia, and even there are minute pores in our body also, but they are called as the sweat pores, through which the sweat comes out. But these pores are different in case of phylum Porifera. They are called as ostia, and through this ostia the water enters. There are several number of ostia all around the body surface, and there is only a single opening at one end, usually that is called as osculum. It is called as the osculum. So these are all coming under the characteristics of the phylum Porifera. Actually we are not supposed to study about the structure of a single animal of a phylum Porifera, but when we mention about the characteristics, we have to mention such terms. So water transport or the canal system is present, which is a characteristic feature of phylum Porifera. And the body surface is having minute pores which are called as ostia. So ostia are minute pores which are severally irregularly distributed all over the body surface, through which water will pass through. And why water is passing through, we have to observe here. So what is the reason for the entry of water through the ostia? And however, there is a single large opening at the terminal end of the branch or a part of this animal, that is called as osculum. So ostia, they are very minute, but osculum is a large single opening. Now inside the body there is also a cavity. So this cavity is called as spongiocele. So spongiocele is present in the central part of the animal, that is the spongiocele. So the water actually enters into the spongiocele. Now what happens after the entry of water? Now actually here the body wall of sponge is made up of two important layers. Outer layer, there is a layer of flat cells which are called as pinacoderm cells, which is not mentioned. But when we go into some little detail about that, the body wall is having an outer flat cells of means which are flat cells, that is called as the pinacoderm cells. And inside into this there is a non-cellular layer similar to mesoglia, some kind of a gelatinous layer, and inner to that non-cellular layer there is a cellular layer, and that is a very important layer. Those cells are called as choanocytes, or they are also called as collar cells. That is a important characteristic of phylum Porifera. So these terms which we have mentioned here, all these are characteristic feature of phylum Porifera. That means we don't come across these terms in any other phylum. They are characteristic only to phylum Porifera. Presence of ostia. So the presence of water transport system or presence of the canal system is a characteristic feature of phylum Porifera. And the body wall, which is having several pores which are called as ostia, that is also another important characteristics of this phylum. And water enters through the ostia, and there is a central cavity called as spongiocele, and also there is a single large opening called as osculum, where the water moves out of this vasculum. So water enters here and passes out through the vasculum. And when water enters here, the inner layer of the body wall contains collar cells which are actually in contact with the spongiocele. In other words, the spongiocele is lined by these choanocytes or collar cells. Now the word collar cells are named because these cells have a collar like structure in them. Now suppose when we draw a cell, at one end of the cell there is some structure like this, which is like a collar, hence they are called as collar cells. So they are normal cells, but in addition to that there is some belt like structure, a strip like at one end, which resembles the collar what we are usually having. So from that collar region there is a cilia or somewhat a flagella like structure, so that is collar cells actually. So these collar cells which are present inside, they help in there is actually division of labor in that region. Some cells help in gathering the food material. So the water which enters inside, the food particles which are present, they are gathered or they are collected by some collar cells, and some of the cells they help in respiration, they absorb oxygen and they conduct respiration, they release carbon dioxide. So that means they do the function of respiration. And even some cells they conduct excretion, they remove the waste materials and send into the and release into the spongocoel, and this cells which are doing the function of excretion, so they remove the waste materials. So like that, among the inner layer or the water transport system, so helps in food gathering, respiration and excretion. So that means the water which enters inside, the food particles are collected by some cells, the oxygen is absorbed by some cells, the waste materials is removed by some cells. So that is why water transport system is very important. And this water where the food is absorbed, where the food particles are absorbed, and also the oxygen is absorbed by respiration, and this water is now added with the waste material by excretion. So that water is carried out through the osculum. That is the important character of this sponges. So the presence of osculum, this system, ostia, spongocoel, choanocytes, collar cells, that is the characteristic feature of phylum Porifera. Then another important character is, now in the body wall, which just I mentioned, the choanocytes which are on the inner side, the pinacoderm cells which are present on the outer side. So in between we just mentioned that there is a non-cellular layer. Now in this body wall there are some crystal-like structures. They are made up of some calcium compounds. So they are called as spicules. So spicules are structures which are present in the body wall. Though they give a kind of support, so they act as a skeleton in the body of that animal. That is the presence of. So these spicules, which are also called as the spongin fibers, they are made up of some calcium material, and they give some kind of rigidity, or they provide a kind of a skeletal framework or support to the body wall and as a to the body as a whole. That is, the presence of spicules is also the characteristic feature of Porifera. We don't come across spicules in any other animal other than phylum Porifera. In addition to that, the phylum Porifera is also having different types of reproduction. There is a sexual reproduction and asexual reproduction. Now this asexual reproduction takes place by fragmentation. Now fragmentation means it is a kind of regeneration, the capacity which has been expressed by regeneration. Suppose if any part of the animal is cut off, is separated accidentally due to some waves or due to some disturbances in the water, due to water currents or like that, or maybe due to some any other reasons, so that fragment or that separated segment, it does not die, or in other words, it does not get destroyed. Instead, the cells in that segment or the part of the body, they will start dividing, and when they start dividing, so these cells, they go on developing and develop into a complete animal. That means the body is capable of developing into a new organism when a segment or a fragment of the body is separated. That is the phylum Porifera. And then fertilization. The animals are bisexual. That means they are called as hermaphrodites. So usually the animals which are bisexual, they are called as hermaphrodites. So male and female gametes are produced in the same animal, and the fertilization takes place inside of the body. So there is, it is called as internal fertilization. So the gametes which are produced, when they fuse within the body of the animal, it is called as internal fertilization. So here the fertilization is also internal, and after fertilization the development is indirect. So here we come across direct development and indirect development. So the zygote, the fertilized egg, sometimes it gives rise to directly into the young one or the individual. It is the direct development. But in some animals the egg will develop into a stage, an larval stage, an intermediate stage, and then that larva will develop into the young one. So this is indirect development. The fertilized egg does not directly develop into the young one. Instead it develops into a intermediate stage, a larval stage, and then the larval stage will develop into the young one of that animal. So in sponges the development is indirect, and that is the characteristic feature of the phylum Porifera. Then in this we come across different examples like Spongilla, and there are Sycon hyalonema, which is called as the glass sponge, where the animal appears like a transparent, it is transparent in nature, and hence it is called as a glass rope sponge. It is called as Spongilla, Sycon, and various Euplectella and other animals are the examples of this phylum Porifera. So these are the important terms which we come across in this phylum Porifera. Hermaphrodite we come across in some other phylum. Presence of both male and female is called as hermaphrodite, bisexual. Then coming to the next phylum, phylum Coelenterata. So phylum Coelenterata is an animal which is having developed than phylum Porifera. So in phylum Coelenterata we come across the word diploblastic. So these are diploblastic animals, but there we don't have come across such arrangement. So in Coelenterata, the diploblastic and as well as the animals are radial symmetrical. That means which we can divide into two equal halves in any plane. But here these are asymmetrical. They are neither diploblastic nor triploblastic. So diploblastic animals and radial symmetrical are the characteristic feature. And here we come across different examples like hydra, jellyfish, then corals, Physalia, all those are examples. Now just one example, Obelia is also another one. Suppose taking the example of one animal and studying about these characters. Now this hydra is having tentacles like this. So there are about 6 tentacles in the hydra. The number of tentacles vary from animal to animal. Now this in this animal there is only a single opening. Here we come across incomplete digestion, but in sponges we don't have such kind of. So water enters into one opening and water comes out through another opening. But however we don't refer as the word as mouth and anus here, because there are several number of ostia all over the body surface, isn't it? But there is no any single opening here. However, there is only one opening through which the water is sent out carrying the waste materials. But there are several number of ostia through which the water enters into the body. Here there is only a single opening, and here which is usually referred as the mouth or just an opening, and this anterior part of the body is slightly raised, and it is called as the hypostome. So in almost all animals we come across this structure. So that opening is present on a slightly raised surface, and thus there the mouth or that opening is situated. Then from this the tentacles are originated. So the number of tentacles vary. The animals may be free swimming or sometimes attached to some substrata. We find both type of animals, marine animals as well as freshwater. So usually hydra is freshwater and most of the animals are marine. Then another important characteristic of this phylum, on the surface of the body, so as it is diploblastic, that means ectoderm, endoderm is there, mesoglia is present in between. Now in the ectoderm region there are some structures which are called as cnidoblast cells. They are called as the cnidoblasts. So these cnidoblasts, they are called as the cnidocytes also. Here C is silent here. So these cnidoblasts are characteristic features of only this phylum. Just like the collar cells which are present only in the phylum Porifera, we come across the phylum these cnidoblast cells which are present only in the phylum Coelenterata. So cnidoblasts, they are one kind of cells which are present here. Now these cells, they are having three important functions. So those three important functions are, so they help to attach this animal to a particular region, to a surface. So they help for anchorage, for proper grip of the animal to attach them. And also these cells, they help in a defense mechanism. So they are having a kind of stinging mechanism. They cause a certain kind of harm to any other animal which comes to attack the animal of phylum Coelenterata. And also it helps to capture the prey. So if there is any single and a small animal here, so this prey means that animal which is to be caught. So it is actually made unconscious by the help of the cnidoblasts, and then it is taken by the tentacles into the mouth or that opening. So that is the characteristic feature of the cnidoblasts. So the presence of cnidoblasts or the cnidocytes, they are the characteristic feature of the phylum Coelenterata. So here there is this what is called as the coelom, and the cavity which is present inside is called as gastrovascular cavity. So there is gastrovascular cavity just like spongocoel in case of the phylum Porifera. So this gastrovascular cavity is again a cavity like spongocoel. So food enters into this, and how the collar cells help in digestion. Here also the cells on the inner region, that is the endoderm. So the endoderm cells are also having the division of labor. So the endoderm has no any other particular name for that. It is a general endoderm layer there. Within that there is division of labor. Some of the cells help in digestion, some of the cells for respiration, some of the cells for excretion, and the waste is thrown outside into the cavity and sent out through the this pore. Now this digestion also takes place in this cavity. It also takes place inside the cell. So that is why digestion is intracellular as well as extracellular. So both the type of digestion we come across in this phylum. But in phylum Porifera, it is actually intracellular. That means some of the collar cells they involve in the digestion. But in phylum this Coelenterata, some of the cells they conduct digestion intracellular. Intra means within. So within the cells digestion takes place, and such cells of course they are having a particular type of enzymes which help in digestion, and extracellular means the cells also release certain enzymes where the food which is present outside in the gastrovascular cavity, there also the food is digested, and the undigested food is sent out from the body. That is how the digestion is extracellular and as well as intracellular. Then another important characteristic of this phylum is the body form is of two types, which are referred as polyp type and medusa type of body forms. Now what is this polyp and medusa? Now polyp means usually these are sessile forms. For example, hydra is a polyp, so it is attached and it is not movable, it is not moving from one place to another place. Such are called as polyp type of body forms. Now medusa means they are usually umbrella shaped, and they are having the tentacles which are on the suspended on the lower side of that umbrella shaped body, and these animals which are having medusa form of body, they are moving with the help of the tentacles. Though hydra is having tentacles, it is also having the capable of locomotion, but occasionally it will move only in necessary conditions. Suppose if it is in search of food, or suppose if it wants to escape when it is facing some danger, so there in such conditions it will move from one place to another place. That is in case of polyp. So in medusa, for example, jellyfish, Physalia, they are having a certain umbrella resembling body, and on the lower side of this umbrella shaped body the tentacles are suspended, which are again of different types, or and they are many in number. So the help of the tentacles the animal will move from one place to another place, so that is called as medusa type of body. So this medusa, which is umbrella shaped in case of Physalia and also jellyfish, so these are the characteristic feature of the phylum Coelenterata. So presence of two body forms, polyp and medusa, is important to this, and nidoblasts, then presence of gastrovascular cavity. This is common in some other phyla, and even diploblastic radial symmetry, which we come across in the other phyla also. But when we speak of Coelenterata only, then it is these terms which are restricted to that. They are having polyp medusa type of body. Nidoblasts are very important. So these nidoblasts are specialized modified cells on the outer body surface, and they are having three important functions. They help in anchoring the body to a particular surface. They help to hold that body to have a proper grip of the body in case of sessile forms, and they help in this digestion, means to capture the prey. That is the function of the nidoblasts. So nidoblasts, they also help in defense. They protect the animal from the enemies. Suppose an enemy or animal comes to attack, these cells, they cause a kind of a stinging nature, and thus they act as a defense mechanism. So under this already I have mentioned the examples, that is the hydra, obelia, then this sea anemone, Then Physalia, jellyfish, all those are the examples of phylum Coelenterata. Then phylum Ctenophora is another small phylum which are commonly called as sea walnuts, or they are also called as comb jellies. So these are the common name for the animals of this group, which are belonging to phylum Ctenophora. Now Ctenophora are having some common characters of Coelenterata. They are diploblastic and radially symmetrical, and they are exclusively marine. That is another important character of this. They are exclusively marine in character. But these two we find both marine and freshwater. Marine and freshwater means they are found in water, but both kinds of aquatic systems. In case of salt, sea water, and in case of fresh water also we find. But Ctenophora are exclusively marine, means we find only in the sea ecosystem, and that is one important character of this. And they are diploblastic and radially symmetrical, just like Coelenterata. They are having digestion extracellular and intracellular. And other characters which are having in this is, now the body surface, they are having 8 rows of ciliated plates. So these combed plates, means just like a comb, these plates are present. So from these comb plates the cilia originate, and these cilia help in locomotion. So such kind of cilia are in 8 rows. That is the important character of this phylum. So that is why they are called as comb jellies. So because of the comb plate like structures which are present, and these are arranged in 8 rows on the surface, and that helps in the locomotion. And digestion is intra and extracellular both. And another important phenomenon is these animals exhibit a phenomenon which we call it as bioluminescence. So they release a kind of light radiations which we can see only in the night time. So that phenomenon is called as bioluminescence, which is actually an enzyme activity which we can see by the help of an enzyme which is called as luciferase, and that enzyme releases a kind of light emission or radiations, and that appears as the presence of light. This is called as bioluminescence. So emission of light by the living organisms, that is bioluminescence. And as usual there is sexual reproduction in these animals, and also the fertilization is external, means the fusion of the male and female takes place in the outer part of the body, and that is external fertilization. And in this we come across various examples like Pleurobranchia and Ctenophora, or the Ctenophora and Pleurobranchia are examples. So this exclusively marine, 8 rows of ciliated comb plates, and phenomenon of bioluminescence are the 3 important characters which we can mention. So the other characters also we should mention, like diploblastic and radially symmetrical, digestion extra intracellular, both takes place here also, and they are common. So fertilization is present and it is external, but here there is internal fertilization. So that is about the 3 important phylum which we come under phylum this animal kingdom. So the next phylum actually is phylum Platyhelminthes. So that I will be taking in the next part.